pueden tener la traducción. I was just saying for Spanish because that they can have translation.
Yeah, ready?
Yep.
Yeah. Right. Hello, everyone, and welcome to the Scaling AI for and across biodiversity, data collection and curation in AI-driven biodiversity research. So that is just the longest title in the world, but it's we have lots of very, very interesting presentations today about AI and how we're using it across many, many different collections. Ariana, would you like to introduce the team?
Thank you, Ben. So we're quite a large team today, so I'll read everyone's names and then everyone can put their hand up if they're in person. So there's myself, Ariana Salili James, Ben Scott, Sanson Poon, Nikki Nicholson, Kit Lewis, Rakayu Harding, Mikael Toma, David Williamson, and Vuta Koch. I think I've got everyone there. Excellent. Can all the in-person team put your hands up, please? Ben? Excellent. Awesome. So if you do have any questions, please come up to us as well. This is the first of 3 sessions. And a little housekeeping, I suppose. The talks are all 10 minutes long. We have a nice sign here when you have 2 minutes left. So when you have 2 minutes left, please make your way to the finish line, essentially. And questions will be left until the very end. So the questions will be right at the end of all talks during the discussion. We are having a quicker introduction so we can have a longer discussion at the end. And I think that's it. Use the translation barcode if you want translation, but all talks for this session should be in English. Yeah.
Cool, thank you very much. And I'd like to invite our first speaker to the stage, Carly Baptist from Conservation International. Thank you very much.
Hello, everyone. Just get slides. Welcome. My name is Carly, and I'm the Nature Tech and AI Innovation Manager at Conservation International. So we work really to build technical capacity across the organization and help teams utilize different technologies and increasingly now AI tools to best advance their conservation work. So what I'll be talking to you about today, is a sort of prototyping exercise that we took on to figure out, that's not the first slide. Yeah. Okay, fabulous. Great. Cool. Is a prototyping exercise to figure out essentially how well AI agents can help with conservation decision support. And oh God, okay. All right, fun with the clicker. So we all know that there is growing demand for nature positive action, but it is really hard to get to understand what technologies you need to use, what frameworks you should be reporting on. And so stakeholders across this landscape are overwhelmed, whether that's on the ground practitioners figuring out what technology they need to monitor an area, or a corporate who's trying to figure out what framework amidst the alphabet soup that they need to follow. So there's this really rapid expansion in the nature tech space, particularly, and it is quite overwhelming for many people. So there's all of these different, external tech providers and organizations working in this space. And one particular use case that we are kind of trying to get at here is how we can use AI to navigate what is a very overwhelming nature tech landscape. So we're doing this in combination with Nature Tech Collective and Earth Genome. And with funding from the McGovern Foundation. And the first kind of test case we wanted to use on how to develop an AI agent to help with these decision-making support is with nature credits. So we're working with the CI Peru team to understand how we can get nature credits for Yaguas National Park because it doesn't qualify for carbon credits. It's a really difficult place to get to, and it's really massive, like 2 million hectares. So it's hard to figure out how to do, a biodiversity baseline, what sort of nature credit framework amongst the, 60 different ones that are out there, and what technologies are needed for monitoring. So while we started with this kind of as a specific use case, really the underlying decisions on what technology to use, what framework to use could be applied to a number of different use cases. But we were really interested in kind of figuring out how we could get an AI agent to answer good questions. So for tasks like which framework or which technology, we put in a number of different inputs into a RAG framework, so retrieval augmented generation, which is a type of AI kind of workflow, where you're using an AI tool, but you're constraining it to particular knowledge sets. And so here we gave particular documents around nature credit principles, the different methodologies themselves, and had the AI tool reason over these and kind of pick particular tools to to follow for particular questions that a user had. So it's the tool pipeline that is kind of triggered depends on the type of user question. Because we found that it is not enough to just kind of chuck everything in as embeddings and hope for the best. You actually have to give AI the AI agents a bit of an understanding of how to reason over those documents. So a lot of trial and error on the engineering front to figure out how you actually get an AI agent to give good answers that are beyond what, ChatGPT could give you. And so we kind of developed this prototype, again, very much a prototype, very trial and error experimentation. But what this actually looks like is, you can give a question, like I want to get biodiversity credits, and it's going to think and give me this table about, which framework I could use, and the different pros and cons of each of them. It's pulling from the framework tool. If I want to ask, what species or habitat indicators I could get, it's going to use this species tool and monitoring tool to give me ideas of particular species or types of indicators I should think about for monitoring. And I can, turn this into a monitoring plan as well. So it pulls from GBIF and IUCN APIs to understand what species are in the area, which ones are endangered, etc. And so we did a lot of user testing and side-by-side testing of, how this tool compares to just asking ChatGPT these answers. And we find that this is much more specific about its recommendations and gives better answers that are citable and kind of trusted, and that we have verified ourselves from our own work with the CI Peru team in developing this monitoring plan for Yaguas. So we're doing sort of the ground truthing ourselves in Yaguas, testing different technologies and different frameworks, and comparing what we've come up with there to what this model or this agent would have given. And so again, we are really, focused on one specific use case of nature credits here, but this underlying kind of workflow and AI agent system, we think has the potential to scale to help with conservation decision making across a number of use cases. So this is just a small slice in the larger landscape. Everyone is creating agents right now. We're very much in an agentic era. And so what we're trying to do is kind of connect the dots across all the different organizations and stakeholders to help curate the knowledge bases that are necessary to train these AI agents. Because the AI is only as good as the data that you're giving it, right? Which is something we know of all models, but is particularly important. And we're finding that gaps in our knowledge are reflecting gaps in what an AI tool can do. So if you'd like to sign up to test it, please, you are welcome to, very much in prototype phase, as I mentioned. But yeah, and if you're interested in talking about AI agents for decision making and how we can make the best of AI in conservation, please come find me. I would love to chat with you. Thank you so much.
Hello. Thanks, Carly. So we're going to leave all questions at the end so we can stay on time. So our next speaker is Daniel Bauer from Senckenberg. Are you here? He's online. Okay, excellent. Are we ready for the next virtual talk? Perfect.
Hello, I'm Daniel Bauer from Senckenberg Society Nature Research in Germany. And today I will present our efforts to build one-stop for wildlife platform for turning wildlife monitoring data into shared knowledge. Work I'm presenting is a collaboration between multiple institutions. One of them is the Senckenberg Data and Communication Infrastructure team that I am part of, but also our audiovisual biodiversity research group, Darmstadt, the Frankfurt Zoological System. Hello everyone, I'm Daniel Bauer from the Senckenberg Society for Nature Research in Germany, and today I will be presenting our efforts to build one-stop for wildlife, a platform for turning wildlife monitoring data into shared knowledge. The work I'm presenting is a collaboration between multiple institutions. One of them is the Senckenberg Data and Computation Infrastructure team, where I am part of, but also our audiovisual biodiversity research group, the Hochschule Darmstadt, the Frankfurt Zoological Society, and the GeoEngine company. Let me start with a problem a lot of people working on wildlife monitoring data will recognize. Wildlife monitoring systems today can generate absolutely massive amounts of data, and we will see an example of this on the next slide. And often this data is used to do some research, to write a paper about it, and then it ends up being stored on a hard drive in the institution that recorded the data. The raw data is often not published at all. And this represents a huge missed opportunity because this data could be used to enable new research by secondary use of the data. For example, we could use it to support conservation decisions or to train better computational models like AI. But instead, we have all these data treasures sitting at the different institutions. Here in this video, you can see the actual scale of this problem. This is the resulting data from just one camera trap that was deployed over a few months. Now, multiply this by hundreds of cameras deployed across multiple projects and in different regions, and you will see that it is impossible to manually manage this data and to analyze it. So what we need is machine assistants to help us with this. But even if we could annotate everything manually, we have the second big challenge. And that is that we need to find a way to share this data and preferably do so in a fair way. However, today, different projects use different formats for their data. They use different platforms to share it. They use different standards to describe the data. And all this fragmentation basically prevents collaboration and makes data reuse nearly impossible. And this is where AI for Wildlife comes in. The project is a joint effort funded through the Biodiv KI program from the German Federal Ministry of Research. And we are aiming to build a platform that integrates the entire workflow of wildlife monitoring data management. So on the platform, you can manage the data, you can upload the data, you can automatically annotate the data using machine learning models. We have a way to involve citizen scientists to validate automated annotations through a human-in-the-loop process. There is a virtual research environment for combining the occurrence data across different monitoring projects with Earth observation data. And last but not least, we also want to facilitate data publication with the platform. Here's how the data flows through the system. So we start with an initial raw media object on the left, and they get automatically processed by the machine learning models in our platform to generate some initial annotations. So the models, for example, detect fauna, they draw bounding boxes, and they propose taxonomic labels. However, we don't just trust these AI models blindly. Instead, we allow registered domain experts and also trained citizen scientists to review these annotations for us. So they can validate the annotations, they can correct them, or even propose new ones. And these human corrections don't just improve individual annotations, they are also fed back into the system and allow us to retrain and improve the AI models. So every correction made by a citizen scientist can make the system smarter and creates this human-in-the-loop cycle where each annotation can be used to retrain the model and get better prediction on the second run. And finally, everything, the original data, the annotations, and the data provenance gets encapsulated in a fair digital object, and this can then be shared and reused by the entire research community. Now, to make the data interoperable, we need a common language. And therefore, we have developed the Wildlife Monitoring Ontology, or in short, WLMO. This is an ontology to describe observation data, and we use it as the semantic foundation of our platform. And of course, we didn't want to reinvent the wheel here, so we are building this ontology on established standards like SOSA for observations and the Web Annotation Ontology for annotations. The key concepts of the ontology are that we have station setups that describe the configuration of individual monitoring stations within a monitoring project, and these stations produce observations. And multiple observations in the platform are usually grouped into capture events, which can then be described together with all the provenance data of this capture event. So, for example, if an animal triggers a camera trap and it takes like 5 photos in burst mode, that's one capture event that gets grouped together and is described as part of this project in this rich semantic ontology. And this semantic layer is crucial for fair data sharing because it allows for other computational systems to understand exactly what the data means without any ambiguity in the language. And speaking of fair data, in the platform, we are implementing what we call webby fair digital objects together with our crates. And you can think of it this way. The data about a capture event on the platform gets packed in a container that contains the raw data together with all the metadata about this data. And this data gets a persistent identifier assigned to it. Now, on the left side, you can see what the humans see when it visits the platform portal. You get a nice interface where you can see the images of a capture event together with the metadata. However, behind the scenes, the server also ships so-called signpostings as part of the HTTP headers. And these link headers can then be interpreted by machine agents to discover the machine actionable representation of the R crate shown on the right side. And by this, we enable automatic machines to discover, access, and process the data basically without human intervention. And we can use the same PID, the same persistent identifier, to allow access to the data in the human readable view shown on the left side and in the machine actionable data package. For individual researchers, we also provide a way to programmatically access the platform through a REST API. And using this API, researchers can then query for individual data points. They can download datasets in bulk, or they can directly interact with the data on the platform, for example, by integrating the API in an analysis pipeline. And the key here is that together with the fair signposting, we want to provide several different ways to make our data accessible. And this includes both ways for human researchers to access the data, but also for computational systems that can then use the data, for example, for AI training with AI-ready data sets. But we are not just about storing and sharing data. We also want to enable new science on it. And for this, we teamed up with a company called GeoEngine, a provider for WebGIS platforms, to integrate the wildlife platform data into a virtual research environment. And this allows researchers to analyze the wildlife observations stored on the platform across different projects alongside Earth observation data. So, for example, researchers can explore temporal patterns, they can correlate species occurrences with environmental variables, or they can analyze habitat prevalences, all within the same platform. So, for example, here you can see a leopard observation stored on the wildlife platform, and in the VRE, this can then be overlaid with Copernicus land coverage data, for example, to see how different distributions of forests change the species occurrences. Looking ahead, we have three main development priorities in the near future. One of them is interoperability. We are implementing the ChemTrap DB standard to ensure compatibility with the global camera trapping community. This is currently work in progress. The second one is that we are expanding beyond the capability to process images. So in the future, we also want to incorporate audiovisual data, videos, and audio recordings. And the third one is that we are evolving the platform from its prototype to a production level system. And this includes things like a robust user management, scalable processing, and long-term sustainability of the platform. And with this, I'm coming to an end. I thank you for your attention. If you want to find out more about the platform, you can follow the link in the top right corner, or you can scan the QR code. And if you have any questions or want to get in touch, feel free to drop me an email by the contact information on this slide as well. Thank you.
Oh, thank you very much, Daniel. I'd like now to invite Ruta Koch to the stage, please, from the Norwegian Biodiversity Information Center.
Thank you. Yeah, so my name is Wouter. I'm working at the Norwegian Biodiversity Information Center, and I'm gonna give a little insight in how I work with combining AI with human domain knowledge. The kind of tools that I've been working on the past few years all have in common that they're identification tools. And I try to make them as broad as I can so that they can be used by kindergartners and experts alike. And of course, a lot of that goes through AI, hence the session, which is a hot topic. It can also be a bit touchy because it's quite unclear what we mean when we're talking about AI nowadays. Most people think about generative AI, which I think has a role to play. I'm not necessarily a big fan, but I think it has its practical uses. And what I want to do in all of these tools is, of course, prevent the data degradation that can come from using AI too uncritically. And I'm trying to make the tools that allow users to do just that. So I have been working on an AI-based identification app and an identification key format where experts can put in their domain knowledge and users can use that to determine a species. And I think both of these are very important. But one of them is very hip and happening, whereas the other is struggling a lot because we're losing these experts that have that taxonomical expertise. And I really want to highlight that we need to do something about that. So the AI app that I have, it's called Artsorakel. It's a Norwegian trained model. It runs at Naturalis in the Netherlands. And the two main focuses of the app as we've designed it are the low user threshold. There's no cookie, there is no user account or password. Nothing is being stored on a server or becoming an observation. So any kindergartner can use it, which is very important for GDPR and stuff. So, and the other part that we're really trying to do is to always communicate the uncertainty in the identification. People are used to apps making mistakes, but I think it's very important to also clearly communicate the uncertainty of these kinds of apps. And in some cases, like these mushrooms, we're even saying like please don't eat anything based on the app because that would be a very bad idea. The other project I'm working on is the Clavis identification keys. I've developed this. I've published on this during my PhD a few years ago. I used Pokemon in the publication to make sure that I wouldn't have any taxonomical dispute for my toy examples, and I didn't, so that worked. It's basically a formal definition of how to store this domain knowledge for the use in an identification key. And since then, since finishing my PhD, I've been working more on that. I've made an interface for experts where they can put in their knowledge in an editor so that they don't have to work with the JSON directly because nobody wants that. And I've made a first version of an interface for users that is also part of the editor. So the expert can go back and forth, see how it works. And so far the feedback has been really good. They're happy with this platform. So I'm definitely going to go on with that. And I'm going to implement this within the AI app to make it more of a general identification tool rather than just being AI. And that made me think about, can I combine these two in a broader way than just having them as part of the same app? And I realized that they're both quite list-based because the AI app, it takes a picture and it will generate a list of candidate species. Whereas a key, in its essence, is a list of species that you're trying to narrow down to a shorter list of species, ideally of length 1. But this means that we have these lists in common. So I'm trying to link them through that. So the basic workflow of that, the first thing I implemented is to take the picture from the user, put it through the image identification model, which provides a list of species, which we can then pass on to the key to be sorted or filtered on just those species. So the end user will just have to answer 2, maybe 3 questions to find out what the AI couldn't. And I think that's working really well. I've made a demo for it. In this case, a dragonfly. It will recognize that, okay, I have several dragonflies here. I know I have a dragonfly key. Click here to go straight to the dragonfly key, pre-filtered on just those species that are relevant. And you have to answer like 2 questions, and we will have great metadata from that. The user will learn more, will have better identifications. So I really think that's a win-win. And this is also something I'm planning on implementing. The other way is more of the generative approach of AI. As I said, I'm critical of its use, but I think it has some uses, certainly the multimodal. So those are language models that can also look and reason about pictures. So it may be able to look if a character from a key is present in the actual picture that the user provided, or it can look at the illustrations that are provided within the key. It can answer questions from within the key, and it can write a little text about the conclusions, a little explanation. So I tested this with a number of workflows that I thought might be a good idea. Some of them were, some of them weren't. The first workflow I call just the explain workflow. So basically the user provides a picture, the image recognition model will look at it, come with a number of species that could be depicted in that image. We can use the key to get all of the characters that we know to be true about all of these species. So these are the things that we know to be true. And we can send that together with the original picture to a multimodal model, which we can ask, can you see this character and can you assess it? And the assessment we can use as a double check to see if it didn't hallucinate that it saw the wings, but it could actually see the wings, proven by the fact that it could see that they were blue, as an example. With that, we end up with a list of things that we know are true for the selection of species that we have and that are visible within the actual provided picture. And we can use that to generate a text which explains why are you looking at one of these species and what may you be looking for to further identify this by yourself. I also tried to reduce. So again, a picture goes to the model. We get a list of species. And this time we take the questions that we do not know the answers to. So these are the questions that would distinguish between these three candidate species. We give the picture to the multimodal model and ask it to figure out, can you exclude some of these species to get one step further than what the other AI couldn't do? Going a step further, we could drop the initial identification altogether and just give it the key and the picture and tell it to do its thing with the key, try to identify what this is. Final example that I tried is kind of a hybrid approach where you do get the candidates from the first model and you generate a little description of each of the species. So it has this character and that and that and that, and this one has that and that, but not that. And you ask it to pick one. So it will look at it more holistically. And what I find is that the explain workflow works really nicely because we have that double check of knowing the ground truth, and we can arrest the model on hallucinating and discard the hallucinations. With all of the other steps, we don't have the ground truth. We're asking the model to make a decision. And the thing with keys is if you get 9 out of 10 answers correctly, you still fail 100% of the time because you will exclude the correct species. So in my opinion, at least right now, and I think that's pretty basic to these kinds of models, they're too unreliable to make these kinds of decisions. And while it is currently too slow and expensive to run this whole workflow of explaining an identification, I think that is where the potential lies, to use this domain knowledge together with the AI and generate a better user experience in that way. So concluding, I would say that AI does not make domain knowledge irrelevant or any less relevant at all. To the contrary, I think we need more of it, especially to double check AI and to augment AI with. And I really want to just once again highlight that this knowledge is disappearing. We need to store it in a good way. And we might also need to invest in the time that biologists need to become good taxonomists. So with that, I thank you for your attention.
Thank you. So our next speaker is Ayub Mania from NMNH in France and Sorbonne University, and it will be a virtual talk.
As AI becomes more central to biodiversity research, extracting the free text of morphological descriptions of species remains a major problem, especially in botany, where identification keys and morphological descriptions are so rich in detail, yet they lack formal structure. In this work, we present a pipeline that fine-tunes large language models to extract standardized traits from floristic text and also enable species-level comparison in scale. In botany and floristic documents, plant species are documented in a long narrative manner. As detailed as this structure might be, it prevents a computer from extracting, recognizing, and comparing the traits. And also, it prevents the organization of this data into a structural format to save the data in knowledge-based systems like Expert3. This brings us to the following research question. How can we develop automated methods for comparing and analyzing characteristics of different species, including the extraction of knowledge from text without relying on human expertise? To answer that question, we define our problem statement to be like the following. First, botanical descriptions exist as semi-structured free text in floristic literature. Also, as we know, the manual trait extraction from legacy floras is not scalable manually. In addition to that, the lack of structure and comparable data limits the systematic species comparison. And as we know, descriptor-based systems such as Expert3 require strict structured matrices that are difficult to populate at scale from floristic documents and manually. So our idea is the following. We propose a pipeline that is based on the idea that teaching large language models to extract standardized traits will be more reliable if we think of this task as a question answering problem rather than a direct text parsing. And we do that via two steps. First, we will build two fine-tuned models separately, but they will work collaboratively at the end because they are designed to do so. We will look at this method more specifically in this example. So given a botanical species description, the first model that is responsible for generating questions will provide all of the questions related to the trait data expressed in the species description. The second model will be responsible to answer the questions asked on the description. As we can see here, the questions and the answers provide trait data relevant to this specific species and this specific morphological description. By combining the two, we can have a matrix format that will make it more easy and reliable to use descriptor-based systems such as expert tree. Our pipeline is made to handle more than one description at a time because the matrix format makes no sense to have only one morphological description. So given a set of morphological descriptions, at each iteration, our pipeline will have to do the following. The species description will be given to the fine-tuned LLM responsible for question generation. This LLM will give as an output a list of all the possible questions that address the traits that exist in species description. This is stage one. The output of stage one will then be given to the second LLM that is responsible for question answering. This second fine-tuned LLM will be given a description, the morphological description, plus the list of all generated questions. As an output, this model will give a list of the answers or the trait values that are addressed by the questions. The combination of both these steps will help us formulate the output into a matrix format that contains all of the species and all of the traits in all of the descriptions. The species format will give us the ability to compare each species based on its traits, which can then be fed to expert tree. Our fine-tuning pipeline was based on a fine-tuning dataset that we have created. We built it like the following. We selected the set of floristic documents. We extracted all possible plant species descriptions. Some of these descriptions were narrative and very long, and that is why we needed to segment them semantically. We then fed these semantically chunked segmentations to a GPT-based model to generate all possible question and answering pairs. We also had a human to verify these generated Q&A pairs. The human basically verifies if the question is relevant to the context and if the answer is answerable through the context. This gives us as a result a dataset of 16,962 samples for the training data and 1,697 for the evaluation data. The descriptions in the evaluation data was extracted from a flora that wasn't present in the training set. In addition to this, and to evaluate our pipeline, we have defined expert-based evaluation metrics, which were accuracy, completeness, relevancy, fluency, and botanical terminology usage, which basically had a botanical expert, and we asked them to evaluate each predicted sample on each one of these metrics from 1 to 5, 1 being the least efficient score, 5 being the most that we can give. The fine-tuning process was like the following. For question generation, we worked with the same dataset, except that we had as input the morphological description and the questions, such as the model can learn to predict the right questions. We used Llama. For the question answering process, we used the same approach, except for the fact that Llama here takes as input all three: the morphological description, the questions, and the answer. The evaluation process was based on the typical AI automatic metrics, such as BERT, Rouge, Blue, exact match. What's more important for us in this presentation is the expert-based evaluation framework that we have provided. We have given 100 randomly selected samples of data to the botanical expert, and we asked them to evaluate the predicted answers on each one of the expert-based metrics: accuracy, completeness, relevancy, fluency, and botanical terminology. The expert was provided with this spreadsheet, and they filled it accordingly from 1 to 5 to express how good the predicted answers on each one of these metrics. As a result, we got these average scores. And as we can see, the lowest one is 4.48 for relevancy and fluency, which is still relevantly high. We present here a system demonstration of our pipeline. We have built an app that puts in practice the pipeline. In this example, the user gives as input a list of morphological descriptions with their plant species. Here we have 4 descriptions. When the user clicks on extract traits, the process starts. So for each plant species, as we see here, we trigger both stages. The trait discovery that contains the questions about the traits, and then the value extraction is about responding to the questions. And we also have a section that shows the source of each answer, and we can track it in the morphological descriptions. These steps are repeated for each plant species, as we can see, and the number of identified traits varies from one morphological description to the other. After this, we trigger another step, which is header normalization step. This part is to address how questions can be asked differently about the same thing. In this part, we try to normalize these questions to have the same format so that we can have a comparable version in the matrix. After this, the comparison matrix is provided for all species with all the traits and the characteristics and the values. The results can be provided as a CSV file, a JSON, or an expert tree knowledge base, which is a format that we are still currently working on. In this plot, we show the coverage of each trait in the comparison matrix. As a conclusion, AI is becoming more central to biodiversity research. However, that still means that we need to develop methods for structured information extraction for our specific use case. Additions to this application, we will add multi-language support. We will also give the user more hand on the data normalization step, which is the header normalization right before the generation of the comparison matrix. The app will be released as an open source project on GitHub with instructions that will help you have the same app locally on your machine. Also, the inputs will be changed. Instead of having a static input placeholder like this, we will be able to support CSV files as inputs, JSON, as well as TXT files. Thank you so much for your attention. Any questions?
Great, thank you very much. Next up on stage, can I invite Sanson Poon from the Natural History Museum?
Okay, hello everyone. So before I start going to talk about what I want to talk about today, I have a question for you. Who was in Okinawa Tech Week last year? Can you raise your hand, please? Very nice, very nice. Now I remember all of you. So last year, I was talking about an AI lab program that we are trying to run in the Natural History Museum in London. So we're trying to do a lot of AI things to help the researchers and also the collection. So we're trying to use AI to build a gap in between them. And this year, I want to talk about how we apply this AI magic across the country, across the whole UK. So this is my talk today. It's about NHM and DCMS AI pilot program. So it's some government program that we partner like the NHM and the government, and then we run some AI proof of concept AI pilot project together with some of the project partner within the UK. So in case you were not in Okinawa last year, or more unfortunately, you forget completely about my talk last year, let me give you a very brief recap. So the NHM AI Lab is doing a lot of things to try to build a gap in between the collection and research within the museum. So we try to do everything in-house. In-house data, in-house collection, in-house researcher, and in-house AI developer. So we did quite a lot of projects, like different projects are in different stages. Some of them are finished, some of them are in research and development, some of them are in the horizon. So well, not to mention that like one of our proof of concept project become a patent as well in the last year. So there is like quite a lot of demand that we can see within our museum, which also means that very likely outside the museum, outside our own organization, around the country, there's also quite a lot of demand as well. So very luckily, we managed to partner with the government, the Department of Culture, Media and Sports. So that's what DCMS stands for. Together with the museum, we come together like to try to have a collaborative model to try to run a program to help different like organizations to try to tackle their problems using AI. So the program at the beginning, we tell like our partner around like the whole country that like, oh, we're having this program, we have like this like AI pilot things that we can try to like help you to solve your problems using AI. So we received quite a lot of application for this program. And then at the end, we selected 5 of them. So not only because of like these 5 projects are very like interesting, but at the same time, we also want to test this collaboration model. Can we actually apply to like the whole country and also know about like what kind of organization will be benefit more from this kind of program? So instead of just about like how interesting how important their project ideas are, we also consider like different like organization type as well. For example, like we work with like botanic gardens, we work with like national museums that is slightly bigger organization, but at the same time, like we also work with some like smaller museum collection, for example, the Royal Agricultural University, and also some local museum as well, for including like this Lyme Regis Museum, a very tiny but very lovely museum. And also we work with a zoo in London as well. So there's a very diverse set of like size of organization, and also like they scatter around the whole country, including Scotland as well. So very luckily, with the support by the by the government and the DCMS, we managed to like put together a team of 7 very brilliant people, well, including myself, 6 brilliant people, including myself. So 7 of us, like in the team, working together to deliver these 5 projects together. So I'm not going to overload you with like all the details, all the technical things, or all the magic that we perform for different like projects, but like maybe I can briefly go through the 5 projects that we have done. So first is the Lyme Regis Museum, that we try to use AI to help them to transcribe like all these like index card. So we managed to develop a pipeline, AI pipeline that can like reduce the time by like 98%. So instead of like just taking a picture of this and then we need to find someone to like manually type all the data, all the information to the computer, we can just like scan this kind of card, and then immediately we can extract all the data that is written in the card. So this project not only apply for Lyme Regis Museum as well, because this type of index card is actually very commonly used around the country for the last like 20, 30, 40 decades. So since like with our help of this like AI tool to pipeline this process, together with the pipeline that they set up in the Lyme Regis Museum to scan all this card, we can actually apply all this kind of work way beyond the Lyme Regis Museum. So at the moment, they are already like asking like all the like other local museum around the country, also like using this card to like record their collection, go to visit Lyme Regis Museum, scan all their card, and then also apply our AI model to help them to transcribe all the things, all the information in there as well. So this is something that actually is a very good example showing that like by just like one AI model that can help us to transcribe this card, not only one museum will benefit of it, but also other museums, other collection, and anyone else pretty much using this type of card to record their data. And then another project that we do is very different from the first one that I just mentioned. It's a project collaborated with the Royal Agricultural University. So I'm not sure if anyone here like have experience with the Charles Darwin's chatbot. So it's like you can ask Charles some question, and then it will try, he will try to like respond you using his own way of like his own language, his own knowledge and stuff. So in here, we try to build something similar to that as well. It's a chatbot of this Professor Woodward. Professor Woodward is one of the founder of the Royal Agricultural University during the Victorian time. And then he has a very unique collection that is hidden in the archive of the Royal Agricultural University. So after sitting there for like hundreds of years, pretty much not many people have access to that. Is this a very good way to try to unlock the collection? For example, like for public engagement, like people can come to the museum, come to the university, try to talk to like Professor Woodward and ask like, okay, for example, what is your favorite plant? And then we, according to what we know about this professor, it can try to answer you as well. And at the same time, like the university can use this as an education tool for their student to try to get a better understanding about the collection as well. And then the third project that I want to talk about is a project together with like National Museum Liverpool. So this is a project that's trying to like generate alt text for like their collection. So National Museum of Liverpool, they have a huge collection. And at the same time, they have a lot of their collection that is already have an image that is digitized and put online. But at the same time, like there is a group of people that would be difficult to try to see this image because let's say, for example, maybe they have difficult to see, difficult to like trying to understand what is like on a monitor, all this kind of thing. So that's why like alt text can be very helpful for those specific group of people. So this is a project that we work with some blind and visually like difficult user to try to build something, try to build an AI tool that can just by like looking into the image to describe what is in the image. So at the moment, the National Museum of Liverpool, they have to do this kind of things by hand. That is not very convenient. But with an AI model, we can streamline this process and generate this kind of alt text pretty much for the whole collection. But at the same time, because like of this kind of like image, not all of them are as easy as describing this is a piece of cloth, but also some of them can be cultural sensitive. Some of them can be like quite difficult to explain as well. So try to use AI to get a balance in between them. And then we are deploying this kind of like model to different like use case. So there's two more projects that we have done. I'm not going to deep dive into that because my colleague Ariana will go to talk about a project with Edinburgh tomorrow. And then also another colleague of mine, Evelina, is going to talk about a snake project that how we tackle like wildlife trafficking using AI later in this session as well. So yeah, they are finished. They're ready, like they're preparing for like publication. Very soon you will see all this kind of like results in the public domain. So this is not my own effort on that. It's a whole collection of like peoples who are like working together to make this happen. So special thanks to the team, like especially Ariana and Ben in the project team, and also all our project partner, and also of course, where's the money come from? Department of Cultural Media and Sport. So I think I will stop here and then, yeah, thank you.
Thank you. So our next speaker is Kenzo Melville from Ghent University. So round of applause for Kenzo.
Okay, hello everyone. Today I will talk a bit more about some automated methods to determine the scale of herbarium sheets. Okay, it's working. So as you all know, in the past couple of years, there has been a huge effort in digitization. This has led to tens of millions of herbarium sheets and other specimens worldwide. And more recently, there has also been work on AI methods on how we can detect and measure traits of these specimens. But crucially, most of these methods, they detect them in the pixel level. So we get these masks or these measurements in pixels. And then we will need to use the ruler, which is usually included during digitization, to convert them from pixels to centimeters, millimeters, inches. And this way we can aggregate them on a large scale and then get some nice research and conclusions about the local and historical biodiversity. However, these methods are often fail in the detection or the conversion of these measurements. So that was the focus of my recent work. So how can we actually do this? Well, we can just simply annotate each tick on a ruler with a point and then calculate the amount of pixels between them. And then we know a conversion between centimeters and pixels, for example. This does assume that the ruler is straight, because we assume that every 1 centimeter distance is the same. So this is a main limitation of the technique and of most methods who do this automatically. To then evaluate the method and also work on it, I constructed a data set where I annotated around 400 sheets from a lot of different institutions. These were taken randomly from GBIF, but also from data sets like PlantClef and others. And each ruler was annotated with its sticks and also the measure. So if it was in centimeter and also how much, because we have these special cases like these lines, which only have 2 points, but these are 5 centimeters apart. As you can imagine, it's quite hard to put dots pixel perfect, and every measurement will have some error. So we calculated basically the deviation in distances for each ruler, and then made it a percentage, and then we can visualize them on a graph and calculate like the uncertainty or the annotation error. And this comes out to about 0.8%, which is quite okay. But this does also mean that using this data set, any method that we develop can not be more accurate than that. We also see that this conversion factor is linear with the resolution, which is what we expect. The higher resolution image, the more pixels that there will be inside 1 centimeter. And then we also noticed that there was a higher error for the centimeter rulers, which is also expected because there are more measurements and the distance between them is shorter because an inch is longer than a centimeter. So in general, there are many challenges. This is just a very small selection of rulers, but there are like thousands of possibilities. Every institution uses a different one or has a different logo, different metrics. Some are in centimeter, inches. But what we do know is that many of them are often reused, which is crucial for the method I will discuss. And current methods, they basically either classify the ruler directly, so they predict which type it is, or they like binarize it to essentially predict the location of the tick marks directly. But this has the issue that if you add new rulers to your data set, you always need to retrain them. You also need to balance them, so you need many examples of each ruler type. And if you just take a random sample, then this is very unbalanced. So we try to do it in a different way to not directly classify, but either match with our known data set and do it that way. So our method will first detect the ruler on the page, and then we basically get the most similar ones from our ground truth data set. And using those, we can do what's called key point matching and try and find a transformation between the two images. And this way we can use our ground truth annotations in blue. This is for the query ruler. So in practice, you don't know them. Then the middle one is the matched ruler from the ground truth set. And then we can detect the key points between both of them and transform our ground truth annotations to the matched image, which are visualized in red. And we can also calculate the error which we made. So we do two types of matching. On the top, it's just image to image matching using clip embeddings, a very popular model. So we just give it an image and ask our data set for the top 3 most similar ones. Then using those 3, we can do what's called key point matching, which basically finds matching pairs of key points between the 2 images, which are similar points. This is a good example. This is of the same monument taken from a different angle, and such a model can basically find the same locations in the image, which were taken from a different angle. And if we have this matching of points, it is possible to calculate a transformation from one image to another image with what we call a homography. So now for some results, we evaluated it on our own data set, and we also got access to part of the data set of Leaf Machine. Maybe some of you will know the model, but they also annotated rulers. Crucially, they did annotate them with only the tick marks, while we annotated the entire object. This also resulted in a little bit of differences in error metrics, but we will investigate that further. We did find that for many of the rulers, over 90%, there is less than 1% measurement error, which is quite good. A couple of them also did not find a good match. And if we look at the mean and the median errors, they're also quite good. They are even better than our human annotation error. So it means that this method is more accurate than I am at drawing points, which is pretty funny. Then we also see, if we look at some results, it can handle overlapping objects, which is good. And I hope it is visible, the dots from afar. So again, the blue dots are the ground truth in centimeters, the orange ones are the ones in inches, and then the red ones are what we predict. And then in brackets, the error is measured. Something that also typically works are partial predictions. So if part of a ruler was detected or only part of it was on the page, because this key point matching method can deal with such cases, it can also map part of the ground truth onto our ruler. So we can still do a prediction in those cases, which you can see nicely on the ruler on the right side there. But of course, every method comes with some limitations. The main ones is that when part of the ruler has changed, like in the top right, the logo is on the right or on the left, but everything else has not been flipped. This messes up this transformation. Also, curved rulers can cause issues, and rulers with not many features, like those lines, it's quite hard to get an accurate key point matching there. But also, not only herbaria use rulers, there are many other data sets which use them, and those featuring more like 3D objects will have even more challenges, because the picture is not taken from above, but might be taken from an angle. So, and also the depth is kind of impossible to estimate, but still, I am hopeful that the method will also work on those type of rulers, which we will test for in the future. And then for my last slide, I want to talk about some next steps. We're planning to scale up the method and write up a thorough evaluation. We also want to set up some kind of public API and go a bit more into detail on like the actual ground truth evaluation by using things like checkerboard markers, so we can even measure the image distortion, because every camera also has a distortion which might not be the same all across the image. And so if you are working on image data featuring rulers or calculating traits or trying to get measurements in real units, you can always contact me and I will be happy to collaborate. And yeah, the code and data set is also published, and we hope to improve our methods. Thank you.
Thank you. Next up, I'd like to invite Jack Hollister from the Natural History Museum.
Thank you, everyone. So I'm going to talk to you today about how we are using robotics in our digitisation process at the Natural History Museum. So natural history collections are very important. They can tell, or they can inform on individuals or groups, and they can do this across time and space. They can do this in the past, and they can also indicate to what's going to happen in the future. However, accessing this data, accessing these specimens is not always easy. A lot of specimens are very precious, some of them are large, some of them are tiny, so we can't just let everybody go into the natural history collections. Oh, sorry. Also, the natural history collections are found all over the world. So if you wanted to study butterflies across the world, you would have to travel all over the world. So it's impossible for a single person to go and sort of deal with all this data. So the solution to this is to digitize natural history collections. By digitizing these, all this information can be stored, it can be shared, it can be accessed not just by researchers, but by everybody, whoever wants to look at it. So at the Natural History Museum in London, we have 80 million objects or specimens. This is broken into 12.5 million pinned insects, it's 7 million shells, 2 million herbarium sheets, and lots and lots more different objects. And these objects come with more than just like the specimen itself. It comes with where the sample was sampled from, where the size, who collected it, when it was collected. There's so much information along with just the single specimen. However, getting these specimens digitized quickly is a big problem. We have a small team at the Natural History Museum, and it's still taken a very long time to digitize these manually. So our solution is to automate the process, and it's using robotics. So the current plan is we want to be able to pick up pinned insects from a drawer. We want to be able to place it somewhere and inspect it using different AI programs. And we also want to be able to extract label information. We want to be able to put all this label information, all the data, all the images taken onto our database, onto GBIF and etc. We also want to be able to check for damage. We want to be able to check for correct classification, make sure it's labeled correctly. We want to make sure the label is present. There's so much stuff we want to be able to do automatically, which we should be able to do. So our current setup, we currently have 2 robot arms. I've named them Darwin and Wallace. I've also built a custom camera system for it. I've custom built some grippers. I've built a turntable so we can get a nice 360 view of specimens. And here's a video of it in action. So it's picking up the specimen, it's moving it to our turntable. So we can now take a nice image. We can then rotate the turntable so we can start extracting that label information. We can see if the label information is present. Go, go, go. Yeah, with just this simple turn-in, we can extract the label information, and then we can place it back. And then we can repeat the process from here. So to do this, we use a variety of AI tool sets that I've built. So firstly, it's the label extraction. Here, I've managed to segment the labels with just a few frames. We can extract the labels, we can stitch them together, and then I've been using large language models to extract this information, which we can then start pairing with our specimen. I've then been building a route planner. So this currently is working with these 3. It detects all 3 specimens, it gives them a number, and then it works through each one and then placing it back in the same place. So here it is. It's detected this specimen, and so it's just going to pick it up and add it to the turntable so we can start processing it, and then it will pick it up, place it back, and then start on the next one. So we're also building in some error detection. So a couple of errors I'm building in. So currently there's specimen issues. Some specimens in the collections are damaged. Some are even missing. The one on the right has come off, it's unstuck itself. So I've built in this little error area where as the robot arm will place the specimen and the human in the loop will know to come over and evaluate why it places one in this area. Another type of error we're looking at is label issues, because we want to be able to extract that label information. You can't always see that label, so we've built in a way for it to add a new label when there is an issue with the labels. So there's a couple of issues with the project. To a human, these specimens look nice and straight, nice and easy to deal with. But to a robot, you can see how irregular placed these pins are. Some of them are at horrible angles, some of them in the specimen at different lengths. All this throws the robot system off. You know, we have to be able to pick that up. We have to be able to pick that up safely. And at the moment, unless I can figure out how to get these pins placed completely vertically, it sort of throws the whole system off sometimes. So just to conclude, the natural history collections need to automate the digitisation processes, and we can do this through automation processes like robotics. And I just want to, our ultimate goal is currently to use robotics to digitise 50% of our collections just using this method. And I would just like to reiterate, this is not to replace humans, but it's to augment the current digitisation staff. We don't want to replace people. We just want to be able to help digitisers with their workload so they can work on the more complicated stuff. And that's me.
Thank you. So our next speaker is Esteban Marentes Herrera from SIB Colombia, and this is an in-person talk. Sorry.
Thank you very much. So I'm gonna present the result of my master thesis along with my supervisor Mario Mora and Jennifer Hironduki. So my research question was, how many beetle species are in a particular state province in Colombia and in Colombia in general? This is not a simple question to solve with traditional methods because we don't have enough money or resources to go sampling in every place. And also there are some curated checklists, but usually are at the state province level. So without knowing exactly how much species there are, it's difficult to assess is the currently 3,000 species we have for Colombia are a lot or we are missing more information. So that's the idea behind. So to solve that, I use an approach to CRISDM. That is a methodology used for data science, originally developed for data mining. It consists of 6 steps: business understanding, data understanding, data gathering, modeling, evaluation, and deployment. And you do these 6 steps in a cyclical model. Here in particular, I'm going to talk about these 3 main steps: identifying the variables that are important for beetles, data gathering and model design, and the model performance evaluation and predictions. For the climatic and habitat variables, I did literature research, mostly in Colombia or some tropical countries, of different studies that have studied how different variables affect the distribution of beetles. And I found these 8 variables are the most common found for beetles in particular. Now, for the data gathering process, I started with GBIF annotated data, mediated data, all the Coleoptera data available in GBIF. Next, I crossed my information with some environmental layers on Colombia, coming from the IDEAM, that is the Research Institute of Colombia. and with some elevation model of the NASA. That left me with 300,000 records that I also put with the checklist for Coleoptera de Colombia that are at the family and subfamily level. And with that information, it was my variable response, so I can train a regression machine learning model. And at the end, I get 42,000 records to train and test my model. For particular, the model design, I did all the modeling in Python, in Spider, using libraries scikit-learn and keras. And the models I choose were deep neural networks and random forests that I check in the scientific literature, were the models that are currently being used for things like that. I'm not going to explain all the models here, only to show the best performing model. In particular, it was a deep neural network when I standardized my data, because deep neural networks can be really sensitive when you have data with a lot of variable. So I standardized with a normal standardizer. And I did search for hyperparameters in both deep neural networks and random forests, so I could have the best performing model. But how I got that model in particular? Well, through some mathematical metrics. In particular, I use R square, absolute error, median absolute error, and medium square error. For the R square, you are looking for values near 1 or -1. I got 0.99, and from the other two, you are looking to minimize the error. So the lowest value is the best performing model, and you can see it's the deep neural network. I also want to check the stability of my model with a k-fold validation, that it wasn't just from a subset of the data that was having the same performance. And I check the loss curves. The important thing here is the loss is getting down with every epoch, and at the end they converge the train test and the test data. I did a split of 80 and 20% of the data. Now, for my prediction in particular, I got a regression model that give me a value for each row. But my variable originally was annotated at the family and subfamily level. So I did a pivot table to gather all the possible number of species for a particular family and get the minimum value, the median value, the mean value, and the maximum value for all the families in Colombia and in Antioquia. And after that, I add this information. That gives me a range estimated of species in Colombia between 3,000 and 14,000, and in Antioquia between 2,000 and 9,000, with a mean estimate of 6,400 for Colombia. As you can see, well, it's a really big range, no? So we have a lot of uncertainty. And how I can address this? Well, with the opinion of experts. So I did an expert elicitation in a qualitative way. I sent my results to entomologists, expert entomologists in Colombia, and I asked them to please assess these results at the national level, at the state province level, in general, or in the family they were experts. And they give me some general estimation, if it was an underestimation or an overvalue. You can see here that in the same general approach, different experts give me a completely different result. Also, for Colombia, in general, someone say me it's an overvalue, and from Colombia, in general, over there, someone say me it's an underestimation. So the uncertainty is real. So having a model, AI or whatever model, is not going to give us a final answer without any doubt. But you can see here the last complete list published by Blackwater, like some 50 years ago, predicted 6,000. Well, it's not that bad, but predicted 6,000, and that is similar to my mean value. So I can say that my model is giving me some really good estimates, the real value should be in the higher part of the estimate, taking into account that most of the expert of the entomologists was on an underestimation. And just some final remarks. This reality in biology in particular, there is still a lot of uncertainty in different groups, in particular in these groups that doesn't have a lot of experts or a lot of modeling. So we still need to rely in experts and also in data curated by experts. These checklists that I use are published by Coleoptera Colombia. There are expert entomologists, so we need this data, otherwise we cannot just put general data and generate a model. The other important aspect is this is valuable, not in particular for the entomologists, they know their group, but more for the general public and for the decision makers, so they can see there is a lot of information still missing, that we should invest in research in this group, invest in conservation. And all information in GitHub repository. Here are the references. And I want to thank GBIF for the Graduate Research Award 2025. Thanks to this award, I plan to continue doing the same for the state province in Colombia and trying to do the same with another insect groups like Hymenoptera and Hemiptera. And a special thanks to the entomologists that helped me without paying anything, without with their time, and to the Instituto Humboldt, Coleoptera Colombia, and my university, Universidad Javeriana. Thank you very much.
Brilliant. Thank you very much, Esteban. Fascinating project. And our final talk for this session is Avelina from the Natural History Museum, and it's going to be a virtual presentation, please.
Hello, everyone. My name is Avelina Leong, and I'm a data scientist at the Natural History Museum. Today, I'm really excited to share one of our AI projects, which is automating species identification of illegal snake goods using a computer vision pipeline. And this is a really great project that demonstrates the interdisciplinary use cases of AI and also the kind of collaborations you can do using this kind of technology. So I just want to briefly go through how this project came about. Essentially, it was a 6-month pilot project funded by the DCMS, and we collaborated with ZSL, who has a really large collection of illegal snake goods that originated from confiscations by the Border Force and the Metropolitan Police. And these items are largely unidentified, meaning they don't have any species identification. And this makes things like digitisation and open access for these specimens extremely difficult. So these collaborations with the Met and Border Force also highlighted the need for quick species identification at the border during confiscations, because often they would have no reptile experts at hand. So we posed the question, could we possibly use AI to speed up this process and make an efficient, time-effective, and quick tool to identify the species of these reptile goods? So this was quite a challenging project. It led us to develop this kind of novel methodology to combat these issues. But what is the big issue? Well, for us, we realized very quickly that we couldn't train this model on the snake goods themselves because there just simply wasn't enough of the snake goods data. In an ideal world, you'd want to train your AI model on the data that it will be processing in the future. So training on snake goods would mean that is exposed to this type of data, and when we do apply it to the snake goods in the future, it would recognize it, and the process would be quite seamless. Unfortunately, that was not the case for this project. So the next best thing we could do here was to train the snakes on live snake images. However, as you can see between these two photos, snake goods really differ to live snakes, and this is due to things like processing and tanning changing the shape, color, or positioning of the snake skins themselves. And so we essentially had to make sure that our model only used snake pattern to identify these species and not any other factors, because snake pattern was the only congruent factor between these two data sets. Furthermore, the model would be trained on live snakes, and this would have to generalize to the snake goods. So just to summarize overall, the pipeline had to be trained on the pattern of live snakes, then had to be generalized to the snake goods. So this posed quite a difficult challenge. But before I get into the nitty-gritty of how this pipeline worked, I'd just like to highlight the amazing work that was done across NHM DataCell and the Met to digitize loads of these specimens for us to use in this project. It was a really great collaborative effort, so big thank you to everyone, especially the volunteers who made this possible. Your model is only as good as the data, so I just wanted to highlight this before I got started. So yeah, here is a very brief pipeline overview in the form of a flowchart. There are quite a few components, but what I want to draw your attention to is the two processes that made sure that our model was only trained to look at snake patterns. And those two sections are segmentation, which is in the green box, and data pre-processing, which is in the light blue box. And to expand on that, I'll just briefly go through the steps of our AI pipeline. So firstly, we have a bunch of live snake images. What do? So we trained 2 AI models, and these performed segmentation for us. Well, essentially, you have a load of images with the snakes and then the background. We want to remove the background from the snakes so that when we feed the images back into the model, we would only be looking at the snake pattern, and we would negate any kind of wood or rock or moss in the background. The second step was we took those segmented snake bodies and we split them into boxes. Essentially, what this did was we eliminated the factors of body positioning, body shape, body size, because those factors are negated when you look at snake goods. The third step is that we wanted to train a classification model on the live snakes so that we can classify between different species. And this is one of the most essential parts of our pipeline, because without this, there would be no classification possible. So once we trained this model up to valuation metrics that we are satisfied with, we then moved on to step 4, which is where we did the same pre-processing steps as we did in 1 and 2 and applied this to snake goods. And this made sure that the data that we're feeding into the model is the same regardless of the data set that we're using. Step 5 is very, very, very important. This is where we actually feed that model in step 3, the images of the snake goods, and see, can it actually translate over to snake goods? Can it still classify different species just based on pattern? And then step 6, we do an evaluation, looking at all the metrics to see how well our model is performing. And there's a mention of binary classification in that box. I'll get to that later on in the presentation. So just again, I want to emphasize these key pre-processing steps that allowed us to ensure that the model was only learning patterns. So segmentation. These segmentation models performed instant segmentation, which is a type of segmentation that does pixel by pixel segmentation. This is really well illustrated in that first photo where you can see the snake is highlighted away from the background, and that background even includes that stick or branch that it's clinging onto. So that means that we've trained these models up to a certain point where it can really understand and effectively classify just the snake. The second step, pre-processing. So this is when we have those segmented snakes, we split them into boxes, and we also apply grayscaling. This really will eliminate things like body shape, body positioning, body color. We only want to look at those patterns. And finally, moving on to model performance. So importantly, how well did our models actually perform? So I'm going to split this up into 2 results. The first result is essentially an evaluation on step 3. How well did that classification model classify between snake species when just looking at their pattern? So here I'm looking at accuracy and precision. We had an accuracy of 0.77, which is quite high, and precision ranging from 0.56 to 0.86 across all the species. The lower end of the precisions were generally due to data imbalance, where we just had much more data on some of these species and a lot less on species such as Python and Loris. But with increased data, we would be able to increase these metrics overall. But considering the range of classes that we have here, these are very, very satisfactory results. Result 2. So importantly, how well did this model actually classify when given snake goods instead of live snake images? So here, again, I mentioned a binary classification, which is actually a classification between 2 distinct things instead of multi-class classification. The reason we couldn't do a multi-class classification in this case, unfortunately, is because of data imbalance and data bias. So you can see that blue table at the bottom, we had a much larger number of Python Sebe goods compared to the others, which had maybe 1, 2, 6 specimens per species. So at this point, statistically, it wouldn't be sound to do a multi-class classification. So we whittled it down to a binary classification instead. And here, the overall accuracy was maintained at 0.77, and precision was also at 0.75 for the others classification and 0.79 for the Python SEBE classification. All in all, these results demonstrate that our methodology of trying to use just pattern to classify snake species works pretty well. So the applications of the AI pipeline, what can we actually do? How do we use this to actually combat the wildlife trade? Well, firstly, it would really, really assist in confiscations where there's no reptile expert in hand. You can increase efficiency and cost effectiveness. And this would essentially be done through an app development where we could link this to CITES and the Red List Index so we can quickly understand how severe this really is very, very quickly. So next, we would also want to expand this to other collections. Could we also look at other types of illegal goods, other types of snake skin, maybe in fashion, and also look into fake versus real skins? And of course, across taxa. So we have this pattern learning methodology. Could we use this to differentiate species of birds, amphibians, insects even? And could this even be done for individual ID, which has been done on orangutan faces or the dorsal fins of dolphins? Could we use that for the wildlife trade? So our immediate next steps right now is to really increase the data set that we have. So can we test this model on more snake goods to kind of prove that our methodology really, really works across a whole range of species? It would also be great to include species that are up and coming in the wildlife trade, those that are trending or on the rise. And finally, this would be possible with more funding, more time, more support in this research. So looking for more collaborations to eventually make this into an app. But yes, this is the end of my presentation. Thank you very much for listening. I'm very sad that I can't be there presenting this in person, but if you do have any questions, please email me. Feel free to contact me and enjoy the rest of your day at the conference. Thank you very much.
Brilliant. Thank you very much, everyone. And can I invite all of the speakers or in-person speakers up to the stage now? We now have some time for discussions. So if you come up to the stage, I'll give you the microphone. And then if people have questions, if you hold your hand up, myself and Ariana will run around and give you a microphone. But if you could wait until you've got the microphone before you start asking the questions. But yeah, if you go up the stage, that'd be great.
Well, it's not ideal, but I have another microphone here.
Okay. Yeah. Hi, this is a question for Jack. I'm curious, I didn't get it from your talk, maybe you said it. So if the robot finds the object in an angle, it seems to assume that the needle is quite vertical in these butterfly collections, yeah? But usually in these insect collections, as you showed for some examples, they can be somehow, whatever, distorted and are in an angle. Did the robot then stop, or is he able to compensate this in a way?
So at the moment, it can still pick it up if it's at an angle, but it just throws off the specimen every time you pick it up and pull it up and in another place. But I am currently designing a method to be able to move the pin vertically and then pick it up, but it's still in a sort of design process at the moment.
For the same presenter, are there any current efforts to expand beyond entomology collections to mammalogy collections? For mammalogists, we need one of the most important features are the skulls, and you can have to shrews very tiny or bats or really large ones. So do you know any effort, or probably your team wants to move to that?
Say that again, sorry, a bit louder.
For mammalogy collections, for mammals, for the study of mammals, just like in that kind of field, what we need is the skulls, and they could be from very small, sure, bats, or the way to really large animals. Are there any current efforts for expanding into that area?
Currently, we're just working with the pinned insects, but we are planning on, once we've cracked that, to start moving on to everything really. You know, within the Natural History Museum, we've got, you know, collections of skulls, everything. So the plan is to just use whatever we can really.
Hi. Hi, I have a question for Esteban. Maybe you mentioned it and I just missed it, but what is the scope of your training set? Is it just Colombian occurrences, or did you train it on a larger scale?
It's just Colombian occurrences because at the end I used environmental variables and habitat variables, and I only got those for Colombia. So I didn't use more data outside Colombia.
Oh, one more question about collections, insect collections at Natural History Museum. 10 years ago, or more than 10 years ago, I was privileged and I had a great pleasure to curate Microlepidoptera collection. So I know very well the problems about the pins. My question is, now we are in a conference about the standards. Maybe you could also think about or just would be in the future plans that a little bit to standardize boxes, because the problem with pins is that the boxes are shallow and especially pins that are of European origin, made in Austria, then of course robots can damage the specimens. Just an idea to think about for standardization.
Yeah, something we are considering is to re-curate the boxes. So a lot of the boxes are, some of them made of cork, so like you said, some of them are in different angles. So we plan to, some of the troublesome ones, to literally pick them up and put them in at straight angles in like better, well-developed boxes. So there is that standardization for the future.
So, hello. I have a question for Carly. So you mentioned, evaluations on the RAG framework, and I was just wondering how you did those evaluations, because you said you compared with ChatGPT, and I was just wondering how you went about doing that.
Yeah, so we sort of have like the ground truthed data from the Peru project in Yaguas, where we've already gone through the process of like picking a framework and picking the monitoring plan. So that's sort of like the answer we're looking for, is basically what took us like 2 years and multiple consultants and, yada, yada. Can all of that get boiled down to, something that this agent can give us in a much shorter amount of time? So in some way, it was really kind of a test against what we had already come up with ourselves, which it sort of did. Like it was recommending the same frameworks and similar monitoring plans. And so that was like the ultimate test of like, can it come up with what we came up with? But then we also wanted to test it against different, nature credit use cases. So we have other nature credit pilots at CI in different ecosystems and countries, and to see how well it would do with that, with those. And so that's something we're working on now. But yeah, the goal was to make something that was better than just a ChatGPT answer, right? So it very much was a big trial and error of like, oh, this worked. Okay, let's do that. Oh, wait, no, this is wrong now. Let's switch to that. And so it was very much a experimental kind of thing. Yeah.
Yep, a question for Kenzo. So you have this nice library of rulers, and it seems to work quite well, the detection and measurement from these rulers. But it strikes me that if you come up with a novel ruler, you don't have any way to measure this. Did you consider some sort of more generalized approach where you can take ruler-like things and find the tick marks and so on?
Yeah, so that is indeed the biggest pitfall, but Yeah, ideally, we only need one annotated sample of each ruler. So if we come across a novel one, we can simply annotate one, and then, yeah, in a perfect world, all those types should always match to that one, and it can then be used. But this is the same issue if you try to detect the strips, like you suggested. You will come across a new variant that's not in your training set. So either your segmentation or detection model will fail, or then your second step to calculate the distance might fail. So yeah, it's very tricky in general, but I do suspect that our method will not work for all rulers. So I think we need something above that even to then kind of decide, okay, which method is now more appropriate. Like the ones with the 5 centimeter line, for example, that would be perfect to just, you can just segment the line and you see it's 5 centimeters, or you can detect 5 centimeters using another model, and then, yeah, you're there.
I've got two questions. One is for Esteban. What is the resolution of ADM of your environmental variables data? And if you considered using subsampling based on a digital elevation model to get, for example, the temperature to actually vary it with the altitude?
Well, I use a mix of resolutions because I was constrained by I wanted to use open data, and the EDM has different mixed resolutions for the different variables. So I just kind of put it all together. I know that it can be perfected, and you can, if you have higher resolution, you can do something to do more fine run. But as my research was like the general state province, I wasn't too concerned about these specific resolutions.
Okay. And another one is for Wouter. What do you think is, as the name of this session is for AI scaling, since models like this that are really good for defining species in a certain area, in certain land, does it make sense to where do you think the boundaries should be for certain models? Like if you want to go globally, because I don't think one model can, for now, decide the species globally. So does it make sense to have certain areas where we know that certain model works, and then in the other model works in a different area, or do you think we should use some different metrics, so like models per different taxa or taxonomic group or something like that?
Yeah, so the way our current model works is that it's ensemble of a number of models. I believe there are 8 models that are taxonomic, and we train a foundation model for all of Europe, and then we fine-tune these models for each country. But of course, country borders are quite arbitrary. I think the south of Sweden and the south of Norway will be more alike than the south of Norway and the north of Norway, certainly if you're going to include Spitsbergen. So I'm a believer in regional models, but I think that this can be included as a factor within a model where you provide it with the geolocation basically as a factor rather than just having pixels. Yeah.
Thanks. I also had a question for Wouter, please. So you talked about you had a format in which you can express expert generated keys. The people you worked with, were they building those keys using that format, or were they translating keys that they already had published into your format?
Both. Yeah. So I've had people who, and I've done it myself a bunch of times to test the format as well, where you have a key and you just go through it. The limitation is that they're often paper keys. So you have one path and you will have a very sparse matrix. And we find that keys that are made by experts using this tool from the ground up provide better results, because I find that the ideal key at any given time has about 4 questions that you can pick from, and you will never have that with a pre-existing key unless it's a key in a similar format. And why would you convert it then? Yeah, good point.
Okay. Hi, thank you for the talks. Also, it's a question for you. I really like the app that you can, like the user can improve the assignation of the taxonomy. But my question is, at the end, the app update that thing with the user selection, or I don't know how you can like update the app or the assignation with these users that are giving you like more information. That is clear?
Yeah, right. So what we're the app is basically just to have an identification. So the whole reporting part is elsewhere. My colleagues take care of that. The idea is to have the whole provenance of the identification stored together with the observation. So you will know the user provided this image. This version of that model said it was one of these species. The user then answered these questions from this key, this version of this key, said these things, and therefore concluded that it was that species. And that's going to be very useful information for the people that are validating. We have experts validating these observations, and that's very important because these observations go back to training the AI model in the next round. So we don't want to have this feedback loop of mistakes from the AI ending up as ground truth in the next version of the app. Yeah.
Hello. Okay, I have a question for Kenzo. So a bit of a silly question, but I was just wondering, do you have a maximum threshold where if it's not that similar to a found ruler that you don't come up with a prediction? Or do you just come up with a prediction with the closest match?
Yeah, so it was in my results, but I think for around 3% of the data set, we had no match. And we do have some, how do you call it? Yeah, some thresholds. So basically manually tested some things. And we typically look at, with the key point detection model, needs to be at least 10 matches with a high enough score. But then also for the clip model, you could set a score. And then even in the last part, we do a last check. Is our conversion factor, is it meaningful? If he says like 1 or 10,000, then we also can skip it. But all of these parameters are manually set. And yeah, it will take some more fine tuning to do it. Because we did have, I think the largest error rates were like around 30-40%. So it would be nice if we could also know when we are not so accurate.
Yeah, I've got one more question, and that's for Samson. Question is, you shown an example of a description of an object that is made for a visually impaired. And did you have some inputs from the visually impaired community, or have you tested it actually for on somebody that? How do they receive that?
Yeah, very good question. So that project is not like, so let's say it's not that just we use AI and then generate some like alt text and then immediately we just publish it as they are. So there is a lot of like human in the loop in that process to make sure that like all the text that generated by the AI model are like reasonable, accurate, and fit what the actual user going to be like, what they are expecting. So we actually work with like some of them, and then also like listening to feedback, giving them like different models output for them to choose to see like which one do they prefer more, which one is like less, is something that like they don't like as much. So it's definitely is very, what should I say, trying to get like feedback from like the user, the actual user, as much as possible. So yes, there's like quite a lot of this human in the loop process, and then all this information will, all this idea, all this feedback will feed back to the model, and then generate and train like a better model for the use of this use case. Yes, thank you.
Anyone else?
My question is for Walter. In your presentation, you talk about pictures where you have a single organism could you imagine, or if you already did that, how can work your modern work with when you have more than one organism, like a species interaction or something like that?
Yeah, the current model does provide me with bounding boxes of where it detects these species. So I could segment this, and for the key, it wouldn't matter because you have the species and the user can look at whatever part they need to be looking at. For the AI pipelines that I showed, I think that would make it even worse. But then again, as I said, it doesn't really work to use these multimodal models to identify anyway. So yeah, I think there's nothing in the pipeline that restricts it to being one specimen per picture. Yeah.
Cool. Thank you very much, everyone. If we don't have any more questions, if we can just give all of our speakers a round of applause. They're fantastic presentations. Cool. Thank you. And a bit of a break from the schedule. We've got a couple of minutes left. Vince Smith would just like to make a very quick announcement, if we may, and then we'll go and have some lunch. That's brilliant.
Thanks, Ben. I appreciate I stand between you and lunch, so I promise I'll be very brief. My name's Vince Smith. I'm head of digital data and informatics at the Natural History Museum in London. And as some of you know, we have a very large digitisation initiative starting in April next year. And the headline is "We are hiring." So if you're a data engineer, a software engineer, a data architect, an AI engineer, we at the museum have got some massive scaling to do as we hire lots of new people, and we have lots of roles. Now, some of those roles will require a right to work in the UK, but some roles we can sponsor people. So if you're interested in any of those roles, come and talk to me or any member of the NHM team here. We can fill you in, and then just keep an eye out on the Natural History Museum jobs website, because literally right now I'm trying to fill 14 different roles, and come April we'll have something in the region of about 30 to 40 different roles. This is a £155 million initiative. We'll actually have roles across the UK, but many of them are centred in London. So if you're interested, give us a shout, and yeah, I'd love to talk to you. Thanks very much. Thanks, Ben.
Great. Thanks, Vince. Thanks, everyone, for coming to the session. Hope it was interesting. And thanks very much again to all of our speakers. Right. Have a nice lunch.
Oh, well, afternoon in a room full of emptiness by a freeway. I confess I